Hello everyone, this is Alush, a research scholar in the field of bioinformatics. In this series of videos, I'm gonna speak about how to handle RNA sample from the moment that it comes out of the sequencing machine until the moment we do differential gene expression analysis. So the first question that might come to our mind, why do we need to do RNA Seq? Or what are the applications of RNA Seq? So we can answer this question actually with 4 points. The first thing, when we are willing to do like transcriptomics study, so we are willing to check any kind of RNA that's existed into the cell. So for example, miRNA, mRNA, lncRNA, and many other types of the things. The second thing, when we are doing gene expression studies, so for example, I'm looking for the differential expression of the genes between two different situations. Like here we don't refer only for the disease things, no, we can speak about two healthy cases. For example, we are seeing which genes are different between when we are awake or when we are asleep. The third thing, it's about disease research and diagnosis. So for example, I want to see which genes that they play a role in the meaning of the disease. So maybe in the comparison between the healthy people and patient, we can see which genes are upregulated or which genes downregulated. Again, we don't only speak about genes, we can speak about anything related to RNA, like non-coding RNAs. The fourth thing is pharmaceutical development. So we are willing to develop a new drug, so we need to see how this drug affecting like let's say the cases or the patient, so we can see which genes have been like affected, upregulated or downregulated after treatment with the drug. So okay, I understood the application of RNA Seq, but how I can do so? So usually the steps of application of RNA Seq, especially when we speak about the differential gene expression analysis, we can see it in this chart. So it started from like I do the extraction of RNA and then I send it to the sequencing machine. The sequencing machine will give me the sequence read. So the first thing that I need to do with these reads or sequence is to check the quality of it. So sometimes this sequence maybe have contamination or they have low quality, so I need to check their quality. If their quality is good, I can go further. If their quality is not good, so then I need to see if I can do some alteration to improve this quality. So usually we check the quality through fast QC, and we do we can say the adapter trimming or trimming of the low quality sequence. So when I trim them, I can get a good quality. If I cannot get even after trimming good quality, so then I need to do the resequencing. So the other thing that also we need to take care of it is the adapter. Adapter is just a small sequence that we added at the beginning of the sequence. So the primer can bind to this little sequence and the amplification start. Actually, this step in somehow it becomes day by day more optional, because many of the alignment tools that align our gene, let's say our transcriptome or RNA sample to the reference genome, nowadays they can like detect this adapter and avoid or trim it by themselves. So after that, I got the good quality sequence, so I need to align it to the reference genome to see how much of my gene, how much the percentage of the sequence that I do have aligned to the reference genome. After that, I need to do count readings, which means to see, for example, how many copies of each gene align from my sequence to the reference genome. So for example, let's say in gene number 1 I have 15 copies, in gene number 2 I have like 5 copies, so which means I can say like maybe the gene number 1 is more expressed than the gene number 2. But we need to ensure that this difference is having a value, or because of a mechanism that's happening. So for that purpose, we usually do statistical analysis to prove that this difference is not happening because of luck or a coincidence. No, it has a value and it has a reason. So now I understood the steps of application of differential gene expression analysis, but from where I can start, from where I can get the data. So one of the most famous websites is Gene Expression Omnibus. It's a repository where the authors and researchers submit their sequences. So from there I can access the data. So how I can use this repository? There are two ways. First way, that exactly I am reading the article, so this article will mention the ID. So all the IDs will start from GSE. So by knowing the ID, I can just, for example, let's say GSE 1, 2, 3, 4, 5, 6, something like that. I can check that I can get access to that sequence. No, I am not that much expertise, so what I can do, I can look for specific keywords. So for example, I can write a name of disease, let's say colon cancer as an example, and I can do the search. The moment that I do the search, it will give me the results. So for example, here in my case, I put colon cancer, I got like thousands of studies, and each study will have like the title, description, and many more information, like for example which platform, what type of expression analysis have been done, what's the name of organism. Got like thousands of studies, and each study will have like the title, description, and many more information. So as I said, the moment that I will press onto any study, I will get some more information. So in this case, for example, we can see here the title of study, when these studies became available, what type of organism, it's Homo sapien, what type of experiment, for example here expression profiling by high throughput sequencing is NGS. Then the summary usually is the same of abstract of the article itself, then the overall design, and this what matters a lot, because here we will understand what they have done. So in their case, for example, they performed gene expression profiling analysis using data obtained from RNA Seq of two different colon cancer treated with control and with the drug with the specific concentrations. Again, here I can find the name of the authors, when they did submitted, when the last update they have done, which person that we need to communicate if we need more information, at which department, which university, and so on. So that's a great information, but from where I can get the raw sequence, actually I need to go down until I will find this RNA SRA run selector. The moment I will press here, it will open a new tab, and this new tab will have more information about the name of the project, the type of analysis RNA Seq, from where the cell line have been, from for example derived here from patients, what type of file type I might find. So here I can find SRA and FASTQ. Furthermore, which instrument have been used. So for example here Illumina Nova 6000, what type of library, it's paired library, and cDNA1. And when is this available? It's became just in the March, so just the last month. What kind of source? It's colon cancer, and so on. I will go down again, I will get information about how many runs. So here they did 18 runs, and the size of the data it's 89 GB. I still need more information, so here I can see each sample, so I can see how many runs we do have. Usually we can have like biological replicates or technical replicates. In the case of technical replicates, we'll have many runs for the same sample, but here we can see for each sample there is only one run, so we don't have any technical replicates. Again, the same thing, we can get more information about like the bases, the bytes, the cell types, the experiment. Sure, there are lots of information, and I don't want to go much more further because we still have many videos to get information about that. So in this video I try to give overview about how we can get the sequence. So if I want to get just one thing from this video, just we need to keep this ID in our mind, because by knowing this ID I can get the sequence. So if you like the video, please like, share, and subscribe, and I would like to get any feedback that will help me in improve my next